Hello to everyone joining today's webinar. I, Junaid Tantre, as a co-convener of this event, I'm delighted to welcome a diverse and engaged audience in today's webinar. Your participation enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversation and collective learning as we embark on this new journey. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. And I'm confident that this session will uphold its reputation as a speaker will put valuable insights into this webinar. So let's seize this chance to gain new knowledge, participate actively, and progress our field. Welcome everyone, and I look forward it for a productive experience. We are gathered here today to commemorate the 76th SPSR webinar on Isatin-Based hybrid molecules, a new era in anticonvulsant research, a distinguished event in the series hosted by Society of Pharmaceutical Sciences and Research under the esteemed guidance of Dr. Shashi Alok sir, National President SPSR. The Society continues to play a pivotal role in fostering innovation and advancement in field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse various other programs pharmacy-related public health issues in India, focusing particularly on disease prevention. We make a priority that to recognize and honor pharmacists whose notable contributions drive progress in our field, ensuring steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of the pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It's a great privilege to introduce Dr. Biplab Debnath, sir, Principal and Professor, Bharat Technology, Howrah, West Bengal, India, a distinguished speaker. Dr. Biplab Debnath's expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussions and expand exploration on Isatin-based molecule hybrid molecules, a new era in anticonvulsant research. We are grateful for his engagement and anticipate an enlightening session. It's a privilege to introduce Mrs. Monika Sabharwal, ma'am, as a convener for today's webinar, who also serves as the National Secretary of SPSR and Managing Editor of both IGPSR and IGP. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of a commitment towards the field. We are grateful to have her guidance in today's session. Also, I'm pleased to invite Mrs. Monika Sabharwal, ma'am, to warmly welcome our distinguished speaker, Dr. Biplab Debnath, sir. Monika, ma'am, over to you.
Thank you so much, Mr. Tantre. Hello, everyone. I am Monika Sabharwal, the convener of 76 SPSR webinar, and it is my privilege to welcome you all to today's enlightening session. The topic for this webinar is Einstein-based hybrid molecules, a new era in the anti-conversion research, a subject of great importance in the pharmaceutical research. We are honored to have Dr. Biplab Debnath, sir, Principal and Professor at Bharat Technology, Uluberia, Howrah, West Bengal, India, as an esteemed speaker for today's session. His expertise and the in-depth knowledge in the field will provide a valuable insights into this promising area of research. I am delighted to share that we have received over 1700 registrations from more than 15 countries, which speaks the volumes about the global interest and the impact of this webinar series. Today, we are celebrating the 76th SPSR webinar, a significant milestone for Society of Pharmaceutical Sciences and Research SPSR. Throughout this journey, we have received a tremendous support and a participation from around the world. Your continuous feedback via various platforms has motivated us to organize these webinars on every Sunday with beyond enthusiasm and a very commitment. At SPSR, we are dedicated to uplift the pharmacy profession by offering these webinars on diverse and relevant topics. Our sessions are often curated based on audience suggestions, ensuring that we address the topics of interest and the value. I am pleased to inform you that SPSR membership is now open. By joining SPSR, you became a part of community dedicated to excellence in the pharmaceutical sciences and research. SPSR membership offers to assess a wealth of knowledge, professional growth, opportunities, and a supportive network of experts and the peers. I encourage all of you to visit our official website to learn more. In addition, SPSR award nominations are now open. I invite you to explore this opportunity by submitting your nomination through our website. We are also proud in collaborating with the national and international institutions to foster knowledge sharing and growth. Recently, SPSR has signed several memorandum of understandings, paving the way for the future collaborative activities that will benefit our professional community. We sincerely thank to the SPSR team for their relentless efforts in organizing such webinars. I also thank our audience for attending it regularly, as your participation and suggestion inspire us to work harder and continue to deliver impactful sessions every time. Before we commence, I would like to warmly welcome Dr. Shashi Yalo, our National President, whose visionary leadership has been very crucial in the impressive transformation of SPSR. I would like to extend my heartfelt thanks for the significant contributions made by Mr. Junaid Tantre, the co-convener of this webinar, and the organizing committee members, Dr. Padmani Shukla, Mrs. Swati Jain, Dr. Pradeep Golani, Dr. Sanjay Nagdev, Dr. Saurabh Kose, Mr. Ankur Agarwal, Dr. Payal Mittal, Mrs. Nidhi Mittal, Dr. Khushbu Mishra, and Mr. Rishabh Banot. Their tireless efforts have played a crucial role in transforming vision of this event into a reality. Join a SPSR vibrant community of over 40,000 pharma professionals actively engaging across LinkedIn, YouTube, Facebook, and WhatsApp. Let's connect, learn, and grow with the top tier webinars and the exclusive content. Do not miss out this unparalleled opportunity to elevate your pharmacy practice. Stay updated by subscribing to SPSI YouTube channel and enabling the notification bell icon for instant alerts. Follow our journey in advancing the pharmaceutical sciences and healthcare for the societal benefit. Let us now look forward for today's enlightful session. I am confident that Dr. Biplab Debnath sir talk will be rich in topics, and thank you for all for joining us today, and I hope you will find today's webinar both informative and inspiring. Thank you, and over to you, Mr. Junan Tantre.
Uh, thank you so much, ma'am, Mrs. Monika Sabharwal, ma'am, for granting me the opportunity to present this webinar today. So we'll start now with a short bio of Dr. our guest speaker, Dr. Biplab Debnath. Dr. Biplab Debnath sir is a distinguished principal and professor at Bharat Technology, Howrah, West Bengal, with an exceptional career in pharmaceutical education and research. Dr. Debnath sir has established himself as a leading expert in the in silico synthesis and biological activities of novel heterocyclic molecules. Dr. Debnath sir's research contributions are significant and impactful. He has published 60 plus research and review papers in reputed international and national journals, authored 5 book chapters, published 8 books with prominent publishing houses, His commitment to innovation is further reflected in his granting two patents. And Dr. Debnath sir is a dedicated academician and mentor. Dr. Debnath sir has successfully guided 18 m.pharm scholars and is currently supervising 8 PhD students, fostering the next generation of pharmaceutical researchers. His expertise and leadership are recognized across the academic community as he serves on the advisory boards of several scientific journals and actively contributes as a member of various prestigious scientific associations. Dr. Debnath sir is a sought after speaker and resource person at both national and international conferences where he also chaired scientific sessions. His ability to inspire and share knowledge has earned his immense respect within the pharmaceutical education and research community. His outstanding achievements have been recognized with several prestigious awards, including Excellence in Pharmaceutical Education and Research Award 2024 from Bioequivalence Studies Center, Jadavpur University, and ABS Clinical Solution. Dr. Biplab Debnath sir's remarkable contribution to pharmaceutical research, education, and mentorship continue to shape the future of the profession, making him the prominent leader and role model in the field. It is with a great satisfaction and pride that we declare that we have exceeded 1,700 plus restaurants globally, showcasing an impressive diversity of countries. Significant attendees is noted from the nation include India, Bangladesh, Cameroon, Indonesia, Iraq, Kuwait, Malaysia, Maldives, Morocco, Nigeria, Oman, Pakistan, Saudi Arabia, and Syria and Vietnam countries. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you once again for all joining us today. Let's make this webinar an insightful and enriching experience for everyone involved from all countries. We have our esteemed keynote speaker here, Dr. Biplab Debnath sir, with us. Welcome, sir. Now, your insights will be highly valuable, and I would appreciate that if you could present them in a speech. Over to you, sir. Please, you can share your PPT, or you can start, sir. Over to you, sir.
Thank you. Hi, Dr. Biplab Debnath. Very good morning, everyone. Especially thanks to opportunity for the platform, Society of Pharmaceutical Sciences and Research family, and my dear friends, Jonet sir, Monika madam, and great leadership, Dr. Shashi sir, and all Howrah Koshe, my dear friend, and Swati madam. So today we are going to start the topics of these Isatin derivatives. So now I can share the
It's visible, sir.
yes, this is the visual. So can I start?
Of course, sir. Please start.
Okay, thank you. So Isatin-based hybrid molecules, a new era in anticonvulsant research. Before the start, at first we know the knowledge about the Isatin. What is the Isatin? So basic knowledge of the Isatin is the fused ring of the heterocyclic molecules. What do the heterocyclic molecule? This is the aromatic and heterocyclic. This is the compound in our organic. So heterocyclic molecules of the Isatin. This is the indole derivative. This is the fused ring. Basically, one position is the indole in the hydrogen, and 2,3 position is the dione. So it is called in the 2,3 diisoindole was first discovered or established in the Edmund and Laurent in the year of the 1841. So, as the product from the oxidation of the indigo by nitric and chromic acid. So, Isatin is the endogenous compound. So, Isatin is the normally basic color in the this type of colors. This is the Isatin. So, Isatin, this is the heterocyclic compound. So, various activities of the Isatin. In here, this is the research-based work basically. So we focused in the drug discovery part in the anticonvulsant. You people here is the know the about the epilepsy, but Isatin basically we discovered the anticonvulsant, or our publication is the anticonvulsant related or anti-HIV. So basically Isatin derivatives, this is the indole derivative, the varieties activities. This is the like of antimicrobial activities, anti-HIV, anti-tubercular, all are activities in the Isatin. So Isatin is the fused heterocyclic ring system and bright orange colored dye. This is the show in the color is the orange. So Isatin has been widely used in the drug discovery. For the molecule of the new molecules, the ring is a part of many naturally occurring compounds. Compounds are designed with diverse biological activities. We already described the activities of the biological activities. This is mean of the antimicrobial, this is the antifungal or antibacterial, this is the antitubercular and antidiabetics and all those. So here is the describing the anticonvulsant activity. So Isatin derives are synthesized using two common methods. This is the very old method of the Sender method and Stolle process. So here describing the, this is the Isatin ring. This is the basically already described in the 2,3 indole ring. 2 and 3 position, this is the oxygen and 1 position is the hydrogen. So Isatin, this is the common epilepsy. At first described in the epilepsy. So epilepsy is derived from the Greek word epilepsia, which means to take hold of the seize. So epilepsy is the chronic neurological disorder. You people know that disorder, neurological disorder is the very common nowadays in the worldwide. 10% people are various types of the neurological disorder patients. It is not a disease but a different cerebral disorder, central nerve system attacked, and basically in the GABA transmitter. This is the called neurotransmitter. So about the GABA, how to attack in the or act in the anticonvulsant activity. This is epileptic brain. There is the show in the brain and attack in the GABA, and these forces act in the ultimately in the anticonvulsant purpose. So epidemiology of the this around 1% of people all world time is affected with this neurological disorder. This is the anticonvulsant. About 50 million peoples worldwide in the epilepsy. Almost 90% of them living in the developing countries. Here is not described in the underdeveloped country. Already developed countries, 90% about the 50 million peoples are attacking the epilepsy patients. So epilepsy is the very vital. So overcome this epilepsy. So we people, our team established the in the Isatin derivatives to how to act in the anticonvulsant activities. So according to the WHO, IBE, ILAE, 2.4 million new cases are added in every year. So common causes of the epilepsy. So describe, this is the abnormal electrical discharge of the brain. This is maybe the abnormal electrical discharge or genetic heredity. Brain lesions due to the trauma, some cases trauma cases, this causes the common of the epilepsy. Infection like meningitis and brain abscess, metabolic disorder like hypoglycemia and hypoglycemia. Everybody nowadays know about the hypoglycemia and hypoglycemia. So some common reasons behind the epilepsy, sudden withdrawal of drugs. This is the like that barbiturate and alcohol. Suddenly withdrawal of this type of drugs can also cause the epilepsy. So history of the anticonvulsant drugs development. How to develop anticonvulsant drugs in the worldwide. First started the drugs. This is the year of the 1850. Now is the 2000 and 2024. Now go to the 2025. First drug is the bromide derivative. Then go to the phenobarbital, phenytoin like the diazepam, valproic acid, topiramate, and now we are going to discover this one already patent these drugs. Different drugs is for the anti-epileptic drugs. So you people, everybody knows in our +2 classes, sodium plus channel how to act or b.pharm courses or graduation courses. sodium potassium channel. So this is the how to attack in the sodium channel, how to activate the gate and open the gate and work in the block the sodium channel and work this type of and blocking of the T-type calcium channel. So this is the main function of the different agenda epileptic agents. So mechanism action of the anti-epileptic drugs. How to act in the epileptic drugs. So first know about the how to action of the epileptic drug. Then go to our particular target in the synthetic part. Basically, I am the synthetic background and drug discovery. And this part is the activity part. So in our team, main motto or main activity is the first synthesize the new molecules. Then go to the drug discovery. Or you people know two types of drug discovery. This is the literature based or survey based, this one, or another is the target based. So in the case of the Isatin derivative, this is the literature based survey, because first Isatin drugs introduced 1841. This is the different types of the activity in our publication. Go to our publication. So the different types of the Isatin activity we already published in the anti-HIV activity, and also patent in the anti-HIV, and anti-cancer means the cell line culture, and the antimicrobial. Now we go to the next target in the anticonvulsant. So this is the literature based study. So what can I do in the literature based? First literature survey, or otherwise we detect in the gene database or protein database. You people know in the drug discovery, this is the protein database or gene database. In protein database or gene database, lot of series of compounds or library. There is the, and select the particular activity part. This is the particular target based. So in the case of this study, this our research team main motto is literature based. And finally, after synthesis the compound, particular show the binding mode. Then confirm the binding mode. We are going to particular activity, because toxicity is the very important. If our drug is more active but more toxic, then we are not go to the market for this further studies. So in our main motto, this is the less toxic but more active. Then we are in the study in the drug discovery. Okay, so first generation of the drug of the anticonvulsant, this is the drugs of the first generation. Already in the market, available markets, all drugs. But some drugs have the side effect or toxicity effect or not potent. So we are going to discover, or our team or throughout the world, several teams are actively engaged for these purposes. This is the second generation of the drugs. So this is the all second generation drug of the already marketed or already available in the markets. Means your pharmacy shop or medical treatment purposes. This is the third generation or newer drugs comparatively. This is a third generation anticonvulsant drugs. So what's the problem? Why we are going to study the new drugs or invest the lots of money, or government use lots of expenses already established, but some drawbacks in the previous drugs in the first generation, second generation, third generation. So we have to plan, discover the new therapy or new drug, new discovery. So neurotoxicity and mood changes. Some current therapy cause a lot of adverse effects. Some drowsiness, hepatotoxicity, megaloblastic anemia, and anorexia, GI disturbances, historian, like that. So some lots of disturbances or drawbacks in the previous drugs of the first generation, second generation, and third generation. So we have to plan the next generation of the drug established. So our drug discovery team, synthetic team are actively engaged for these purposes. So problem related to current therapy. Several types of the problem in the current therapy of the anticonvulsant drugs. So vertigo, nowadays vertigo is the very common problems in the drug is not only in the anticonvulsant drugs, various types of drugs we people are used normally. This is the actively problem. So vertigo, diplopia, neutropenia, GIT disturbance, hypersensitivity, skin rashes, anergia, hypotension, megaloblastic anemia, drowsiness, sedation, aplastic, all problem are come in the together, and mood swing problem, memory loss, all are current therapy problem. So in our particular research part, this is the pharmacophoric parts of these parts. So in the case of the anticonvulsant drugs, mainly pharmacophore, this is the active part, whatever any, we already in the study, this is the dynamics and kinetics, already study. So pharmacophore, this is the bulky position or group in the active part of the whatever any drugs, active molecules is needed for the more active or more potent of the drug. So this is the mainly in the hydrophobic domain, hydrogen bonding domain, distal hydrophobic domain, electron donor group. So these four parts is the main active or main potent or main reason for behind the drug discovery. This is the hydrophobic domain. So here is the, this is the structure, carbazamine, phenytoin, all are or our synthesized compound, all are is the just seen is the four groups: hydrophobic group, hydrogen bonding, hydrophobic, electron donor group. So aim and objective our research or our team are going to this type of activity. So more efficacy, less toxicity, more resistance, tolerance, dependency. So synthesize novel Isatin-based molecule for anticonvulsant activity. So what happened? Reduce toxicity because before establish all drugs have the several problems. So we overcome this type of problem. So it's more important how to reduce toxicity. Then we have to plan, synthesize new molecule and check the ADME studies. You people know the ADME and toxicity studies. So ADME and toxicity studies establish the drug likeness properties and molecular weight, lipincy rules, all are correlated parts. So reduce toxicity, enhance potency, then new drug. So plan of work. This is our Isatin-based novel molecule. Actually, basically, this is our research part, and particular whatever any research part, this is the narrow parts, not in the broad spectrum. So we have to focus in the overall parts of the research on how to establish the methods. So here in the describing the how to synthesize Isatin. First, Isatin is the in the market in the available. So Isatin molecules to derived and out compared to in the drug design, converted and analytical part and toxicity part, and finally after long time, you people know whatever any drug synthesize and process, minimum year 10 to 12 years to come to the market. After all studies in the phase 1, phase 2, phase 3 and phase 4 is the post-surveillance studies. So rationale for novel hybrid molecules. So already described the hydrophobic unit, hydrogen bonding, donor group. This is the important for the whatever active parts and more potent. So this case is also same. So in the scheme of our hybrid molecules, because what can I say? This is the hybrid because indolin and benzthiazole, this is the fused and forming the hybridation. This is called in the hybrid. How to prepare it? This is our compound 4A to 4I. This is the one series. How to mixing and DMF, sodium hydride, reflux and ice cold condition. This is the synthetic process of the scheme. This is already established in the good journal, already published in the good journals in the Elsevers. This is the scheme 2. This is another compound established. This is the 5A to 5F series. This is the ring of the Isatin particular molecule and position of the 1, this is 1, this 2, and this is 3. This is the changes of the, and this is the hydrophobic or these positions in the changes. So physiochemical data. So very important, this molecule is really this is our molecule or byproduct. So need to confirm. So TLC thin layer chromatography is important, and melting point, molecular formula, molecular weight, color, yield, and log P value. So we is the established, and this is the finalized, and all our NMR, especially in the 13C NMR, proton NMR, we send the particular and good results, analytical part. So physiochemical data. This one, all our molecules, newly established molecules in our lab. So spectral analysis in the FTIR, we confirm that this is the functional group is the present or not. So in the functional group, we show that this functional group, this new groups, new molecule attached, sodium NH group attached, and this one attached, they prove that this molecule is the, in our molecule is the really this one. So spectral analysis and proton NMR studies the hydrogen group is the proton means hydrogen group is the available or not. So this is the really synthesized and pure formation. All our spectral analysis, mass data confirmation. The molecular weight of the molecule is the, this is the for or extra byproduct is not coming. That's why it's needed in the mass data. All our spectral analysis already described all molecules. These are spectral analysis. These are spectral and confirm the molecule is the proper way. So all are spectral analysis and confirm the FTI report, IR report. This is the your pure molecule. All are established. That's why just showing the. Then go to the anticonvulsant screening. How to anticonvulsant? So this is the two test. This is the PTZ test and MES test. This also confirm the in the our albino studies. This is a confirmation in the 0.5 hour and 4 hours interval. This is a behavioral toxicity screening. This is a confirmation anticonvulsant screening. And whatever best active part of the compound compared to phenytoin and carbamazepine, this is the already established in the marketed compound of the these two, and compare our newly synthesized molecule. So this is the showing the more active compared to the already established the compound. So what happened? Phase 2, anticonvulsant screening and hepatotoxicity study. In these cases, what happened? Some molecules have a good anticonvulsant activity, already described, but have a toxicity problem. Then that's also important. So we need to the toxicity study, hepatotoxicity study. In these cases, it also passes and shows the good results. So in the case of the, now going to the our in the base code, chemistry parts, pharmacophore distance mapping, because we have to establish a confirm in the pharmacophore distance, because nowadays we can't go to the directly in the animal study. First finalized the screening of the pharmacophore modeling or drug discovery. Otherwise, ethics committee not approval for the in the activity part in the animal studies. So prediction of the ADME properties in the, because of the ADME properties, this is the guideline molecular weight less than 500 Dalton. So all compounds are below follow these properties, and Lipinsis rules, not violation of the not a single compound. So all are passes in the ADME properties, prediction of the ADME properties. Now going to the, this is the molecular docking study. This is a very important part of the all are chemist. As a chemist, we know the molecular docking. How to confirm the molecular docking? In the molecular docking part, various software is available nowadays. This is the free version or commercial version. As a part of the drug designing, go to the part in the docking studies. Then in the, you people know in the Autodox software. Autodox software is the available. This is the Biplab software and some commercial version, now the Schrodinger, then Gold, then Cygress, then Flexex software. So we are studying there in the Schrodinger software. Schrodinger is available in our that time in our lab. So you can people also study in the Orthodox software. This is available in the academic version, and good publication is the reported in the industry as usual academic parts. So docking study, we first confirm the series of the or library of the ligand. In the case of in these cases, we screening the 1000 molecule of the library of the anticonvulsant activity. Anticonvulsant activity, we screening 1000 molecule from the zinc database and protein data bank. This is the simply RCSB software. Go to the RCSB and download the particular protein for this particular the anticonvulsant, whatever anti-HIV, antimicrobial, anti-tubercular, or go to the available software, because lot of PDB protein data bank in software is available already. There is the bank. So as a physics or crystallography part, this is the main work for the store the all software or database. So in the particular PDB, we downloaded the PDB, and then preparation of the protein. Protein is this is the macromolecules. If people know, this is the protein, enzyme, all are macromolecules. This is in the protein part. This is some part is the active or some is the inactive parts. So we first confirm based on the literature survey which part is the confirm. Maybe A, B, C, different parts. In the A part maybe confirm, B part or C part, but first confirm or sure about which part is more active for our particular ligand-based compound. So in the case of these molecules, in our ligand-based, so part A is the active. So part A, water molecule, hydrogen bonding, screening and minimized, and confirm, and protein preparation. First protein preparation, then ligand. Ligand, this is the particular ligand means particular molecules. So this molecule, series of molecule, minimize and energy minimization, then go to the ligand protein preparation. Lig prep preparation, called this one, ligand preparation, then docking study, and always come to the docking study result is the negative, because there is the free energy. Forces free energy and come the negative result minus, means negative means minus result. So this minus, more minus, this is the more active. So in the case of the Maestro software, we use the software the Maestro 10.5. This is a Schrodinger Maestro 10.5 program, and in the force field, similarly, extra precision case of the grid score result, extra precision or SP. In the case of some descriptor, hydrogen bonding interaction, electrostatic interaction, hydrophobic enclosure, pi stacking, all parameter follow, and ultimately image will come in the docking score. This docking score is the good docking score compared to already established in the marketed drugs. Compared to this is the good result for our newly developed compound. So that's why we are going to plan to the patent. So in the case of the docking study, best docking study, then we go to the animal studies and finalize the animal study. This is the correlated best animal activity, showing also best docking score and best ADM score. So in the case of the molecular docking site, this like that we normally used in the PDB 70 TD. This is the docking score is the 9.656 kilocalorie per mole. So your case is this is the minus 96, and docking study the 4F compound is the minus 8651. So this is the docking study of the another molecule. This is the binding energy, hydrogen bonding. This showing the hydrogen tyrosine, and this is the 1.85 is the bonding distance. So docking score this case is the minus 9.3, and all compound are docking like that. So molecular docking study is the nowadays very important. Without, as a chemist, we people can't imagine without molecular docking. First, so know about the docking study, then go to the synthetic part, and then finally go to the activity part. So structure activity relationship. Okay, this is the established our compound is the best compound. But SAR study, structure activity relation study, proved that this is the best compound, our 4A to 4I series or 5A to 5F series. So provides the hydrophobic attraction, hydrogen bonding, all are shows the best correlation between this molecule or newly synthesized molecule and showing the anticonvulsant activity. So go to the conclusion. Now go to the conclusion. So we two series indole derivative already described. The indole derivatives means Isatin is the indole molecules. This is the fused ring of the indole. So benzyl acetamide series 4A to 4I and oxidazole amine derivative 5A to 5F are synthesized, and like activity studies follow in the ADME studies and docking study in the normally used in the software, in the Schrödinger software, and show the best results. So synthesis compound were tested for their anticonvulsant activity using two tests of the MES and SCPTZ model. You people know in the animal studies. Among all the synthesis compound, best compound is the 4A, because our showing the docking result, already showed in the docking result. This is the best results of this series is the 4A series, and the activity part also in the MES and SCPTZ both are the show good results. So compound 4A was further evaluated for phase 2 trials, because phase 1 this is the clear, then go to the phase 2 trials and was found to be there were any neurotoxic side effect, because anticonvulsant, go to the first discuss about the anticonvulsant, this is the neurogenic disorder with the correlation. So less hepatotoxic study and any neurotoxic effect, this is the go to the clinical trial phase 2. And docking study, the most potent compound 4 displayed more favorable interaction through GABA binding sites. So future aspects of the nowadays in the artificial intelligence is the very important part. Everybody knows totally world is the key point of the artificial intelligence. So we have to plan in our newly molecule compounds, especially in the 4A, future have to correlated with the artificial intelligence and more shows the different types of activity using the AI help into the career selection for the better ADME properties, better drug likeness properties, better docking scores, and best potent compound in the worldwide. So this is the follow the different references. So we published this one in the molecular structure is the Ilsaver, is the impact factor is the 4. Thank you everyone. This is actually basically research based work and very specific. So few people said first to in about the grow the interest, so know about the basic chemistry of the indole derivatives and docking studies, ADME studies, all are important and correlated part of our pharmacologist. So this is the anticonvulsant part. So totally is the research part and research topic. So thank you, Jonas sir, thank you, Monika madam, for giving the platform, the opportunities. Thank you.
Thank you so much, sir. It was nothing sort of exceptional. And thank you once again, Dr. Biplab, sir, for answering for giving us more detailed about this topic, for delivering such an insightful and engaging lecture on Isatin-based hybrid molecules and new era in anticonvulsant research. So your comprehensive presentation has provided us with a deeper understanding of a potential Isatin-based molecules and their role in advancing anticonvulsant drugs. And the clarity with which you have explained the design, developments, therapeutic significance of these hybrid molecules was truly remarkable, sir. We sincerely appreciate your time, effort, and expertise in making this session so enriching and informative. Thank you once again for your outstanding contribution to the 76th SPSR webinar, sir. We have taken few questions from online that we have received. So one is, what makes Isatin-based hybrid molecules a promising area for anticonvulsant research? In a short way, sir, you can answer them.
Sorry, sir. How to
The question is, what makes Isatin-based hybrid molecules a promising area for anticonvulsant research?
already described, this is the literature-based survey. Previously already established the some activity part of the Isatin-based, because indole, this is the hybrid molecules of the fused ring of the this type of ring. So already established the different types of the activity. Never new in the nothing parts of the anticonvulsant, but in our hybrid molecule established the anticonvulsant use of the drug-like properties, especially in the docking studies. Based on the docking study, then go to the animal activity and show the good result. So come in the best active part of the anticonvulsant activity. Yes.
Right, sir. We have one more question that do Isatin-based hybrid molecules show promise for other neurological disorders beyond epilepsy, such as neurodegenerative diseases?
Yes, yes. This is the because Isatin-based hybrid molecules is the neurogenetic disorder or other part because of the this is the GABA receptor attacking the GABA receptor, and GABA receptor is the part of the neurogenic disorder is the main reason behind this.
Disorders.
So we are going to have the next plan. Our one PhD scholar working for these purposes. We very shortly will disclose this one.
Sure, sir. Thank you, sir. We have, I think we will be closing it with the last question. What are the future prospects for the commercialization of Isatin-based hybrid molecules in epilepsy treatment?
Yes, we have to plan already patent for this particular compound. We have planned to commercialize with the third party, and we will wish for very shortly with the market and available very short of the times. Thank you.
Thank you so much, sir, for answering all the questions so wonderfully and with such a depth and clarity. Your insights have truly enriched our understanding of isotin-based hybrid molecules and their potential in anticonvulsant research. So, your expertise has left us inspired and eager to explore this exciting area for research further. Thank you once again for your valuable contribution, sir. I hope it will be good reference for future coming PhD scholars who are doing research in such area. It will be definitely a helping hand for them, sir. For concluding the session, as we conclude on 76th SPSR webinar on isotin-based hybrid molecules, a new era in anticonvulsant research, we extend our heartfelt gratitude to our esteemed speaker, Dr. Biplab Debnath, sir, for sharing his deep insights and expertise on fascinating topic. The session has shed light on immense potentials on isotin-based hybrid molecules advancing anticonvulsant therapies. Dr. Devnath's clear explanation on structure-activity relationships, mechanism of actions, the role of hybrid molecules in improving pharmacokinetics, and overcoming drug resistance has truly been enlightening. This webinar has not only expanded on understanding, but also highlighted the importance of continued research and innovation in the area to benefit patients suffering from epilepsy and related neurological disorders. We are deeply thankful to Dr. Shashi Alok sir, the National President SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, sir. Thank you once again. I would also like to express my gratitude to Mrs. Monika Sabharwal, ma'am, the convener of this event, for playing a pivotal role in organizing this event. We extend all the gratitude to all our SPSR team members for their collaborative efforts who have broadened our understanding and fostered cooperative learning. Now, we thank all the participants for active engagement and thought-provoking questions, which made the session even more interactive in the end. We look forward for seeing you at future SPSR webinars. We continue to explore groundbreaking topics in pharmaceutical sciences. Our upcoming 77th SPSR webinar, international webinar on drug product development and industrial approach. Our esteemed speaker for that event will be Dr. Saroj Kumar Raut, sir, Formulation and Process Development Scientist, R&D, LNK Internationals, New York, USA. The webinar registrations is given in the description box. Have a wonderful day ahead. Thank you so much once again for making this webinar successfully. Thank you, Devnath sir. Thank you so much for your
Thank you.
time.
Thank you. Have a nice day. Thank you, everyone.
Thank, thank you,
Thank
sir.
you.